being part of the Scripps Institution of Oceanography and listening to my colleagues who study ocean-atmosphere interactions, or people who are interested in ocean chemistry and ocean physics, they frequently talk about ocean observing. I hear them talking about ocean observing and developing proposals and technology to deal with ocean observing. And so, how scientists observe the ocean means everything for what we're able to know about it. Researchers working here in the 80s played a major role globally in discovering the microbial loop and communicating it to the broader scientific community. We are building on that foundational work to study the microbial loop in molecular and genetic detail with the modern tools of biology. But we still use this sampling ladder and go down to the bottom of this pier with our nets and buckets, and take the material back to the laboratory and try to culture isolates and obtain pure cultures of organisms, so we can do experiments with it and with them and sequence genomes and try to understand something about their physiology and ecology. My name is Andrew Allen. I'm a professor at the Scripps Institution of Oceanography and the J. Craig Venter Institute. We are interested in phytoplankton, which are single-celled photosynthetic organisms, perform all of the photosynthesis in the oceans. So about half of photosynthesis on Earth occurs in the oceans, and only the very top layer of the oceans, maybe the top 200 meters, are a zone where there is light availability. So the vast majority of the ocean, the interior, is dark and cold, and all of the life in all of the different parts of the ocean is largely a result of this photosynthetic action that happens in the top 150 or 200 meters where there's light availability. And so what's happening is that photosynthetic cells are converting light energy into chemical energy. So they are able to encode proteins in their genomes that are effectively light antenna, and they will vibrate at the right frequency of wavelength of light and transfer energy into molecules that can store chemical energy. So, to me, this conversion of light energy into chemical energy is endlessly fascinating. And, all of the biology on all of the planet is a result of this light-driven conversion of light energy into chemical energy. And in the ocean, we are interested in the adaptations of phytoplankton to take advantage of light. And in particular, there are other nutrients that phytoplankton require to grow and divide, principally nitrogen and phosphorus and iron and other metals and vitamins, and diatoms require silicic acid also because they make a glass or silica cell wall. And we're interested in the genetic adaptations that different groups of phytoplankton employ to acquire the nutrients that they need to convert light energy into chemical energy. And so we're out here on the Scripps Institution of Oceanography pier, and this is a ladder that was used by plankton ecologists in the previous decades, and they would use the sophisticated sampling technology of a bucket and nets, and go down here and collect the seawater in the bucket, and then go back to the laboratory and see what's growing. What's interesting is that in 1 milliliter of seawater, right there, I think 2.5 milliliters in a teaspoon, to put this in context. And in 1 milliliter of seawater, there are around 1 million bacteria and 1 billion viruses and several tens of thousands of phytoplankton. And they are all interacting with one another, potentially in interesting ways, or competing with one another. And these ecological interactions that promote or discourage growth is one thing that we're interested in. Interestingly, only about half of the organic carbon that results from photosynthesis by phytoplankton in the sea makes its way up the food web directly through grazing by herbivores. So a lot of fish and these dolphins that we can see out here, and these seabirds, right? All of the nutrition is coming from organisms that are eating phytoplankton. But when you measure how much carbon dioxide is going into phytoplankton and then how much is going up the food web via herbivores, it's only about half or less. And so what happens to the other half? The other half makes its way into this very, very, very large pool of organic carbon in what oceanographers refer to as the dissolved organic matter pool. So the ocean has very high concentrations of dissolved organic matter, which contains dissolved organic nitrogen and dissolved organic phosphorus and dissolved organic carbon. And the carbon in the dissolved organic matter pool is consumed by bacteria, these million bacteria per milliliter of seawater. And then other things like ciliates and protozoa and fungi, components of the microbial food web, are feeding on the bacterioplankton, and then they also are getting consumed by larger organisms. So this microbial loop of organic carbon fixed by phytoplankton that goes into the dissolved organic carbon pool that is then consumed by bacteria and shuttled up to higher trophic levels that way has been a radical discovery in oceanography. In fact, in diatoms, which is a type of phytoplankton, biomineralized silica on their cell walls, and they can often be very beautiful, and they seem to have maximized trade-off between being very tough and durable, but also being able to grow very, very fast in the pelagic ocean. And so one thing we have to think about is how are we going to sample plankton. This is often done on ships, expeditions, and classically the oceanography community worldwide has collected plankton ships, and there's actually something called the continuous plankton recorder, which is a large spool of tape basically on transit ships, and this is one way that plankton has been sampled for decades, but there are limitations associated with that because when the material is recovered, it might not be amenable for DNA analyses, or might not be the right size class of plankton. So it's kind of humorous to me that in the year 2021 we're still worrying about and thinking about and improving and optimizing how we sample and observe plankton. And ocean observing in general has just always been a major topic. And in biology, I would say that the last 20 years, of course, have been a renaissance for biology. You could argue that biologists have learned more in the past 20 years than maybe in the previous century. We still lack sophisticated tools for ocean observing and the biology side of oceanography. Physical oceanographers have profiling floats that survey the water column vertically, many times a day, and send data to satellites. Short-term physical dynamics in the ocean are beginning to become clear in a way they never have been before. So event-scale processes are able to be resolved. The biology side of things, we're very envious of this, and we would love to be able to understand more about how cells respond on very short time scales to event-driven scenarios in the ocean, right? And that's something that we're interested in developing tools for. Complexity of microbial food webs requires that we develop better, automated, more high throughput quantitative tools for understanding the molecular response of cells to ocean perturbation and imaging. So we have to do all of that. So let's
go get a bucket.
Yeah.
How old do you think this is? I don't know. I think they redid the pier at some point, maybe 10 years ago, and I think I mean, clearly with ocean erosion, they've had to replace these boards, right?
I always just really get a thrill out of coming down here. I feel like it's such a privilege, and I feel so lucky to be able to come out here. It's really inspiring just to look at biologically what's going on around here. We dip our nets and buckets into the surface layer. seawater here, and then use the sample for microscopy experiments. Also for monitoring harmful algal blooms and the occurrence of pathogens and the response of the base of the ocean food web to atmospheric conditions or other factors that might trigger harmful algal blooms. This resource is a great thing to have access to. This very basic sampling apparatus at the end of the pier is fantastic, and it's one reason why a lot of biological oceanographers like to be at a place like Scripps is to have this kind of access to the ocean. We also realize that we need more sophisticated and high throughput ways to study the complexity of plankton ecosystems. And so there's a lot of emphasis currently on automated imaging technology or automated ocean observing technology. And you can imagine that physical oceanographer is referred to as atmospheric forcing that drives cold, nutrient-rich water from below the thermocline up into the sunlit layer of the ocean, and this produces phytoplankton blooms. And if we are coming out to the pier and sampling weekly or daily, we might miss those events. And the interesting window for really learning about physiology and growth might be, right when the phytoplankton are first observing or encountering the nutrients, and then we want to understand cellularly how they respond to that and how that translates into growth and a bloom and the dynamics. So we need better tools to observe these phenomena, and
like constantly,
right.
Instead of once in
right.
every now and then.
Exactly. There's always perhaps things that haven't been observed before, and there is a famous story of a cyanobacteria called Prochlorococcus that was discovered and has been studied extensively in the laboratory of Penny Chisholm at MIT. And Prochlorococcus was discovered by an instrument that just had an order of magnitude improved sensitivity. And suddenly the most abundant photosynthetic organism on the planet, people began to know about it, and this was in the late 80s. So, right, there's always new things to be discovered, and we need to understand natural responses to seasons and cycles like El Nino cycles and other oscillations that cause variation between years. I mean, there's naturally very productive years and years that are less productive, and we need to understand natural variation in a level of detail and resolution that enables how ocean acidification, which is a result of carbon dioxide dissolving into the ocean. The idea is that there are multiple stressors: temperature, oxygen, nutrient loading, and things like ocean acidification, and all of these things might have an impact on the fundamental molecular mechanisms used by phytoplankton to acquire nutrients. And so it's important that we have a detailed and mechanistic understanding of how phytoplankton acquire nutrients and how those acquisition systems might be affected by environmental change. We often sample plankton with buckets and nets at the end of the pier, but we've also been interested in developing automated technology or implementing automated technology that will lead to higher resolution temporal sampling of microbial communities.
This is so cool. So you guys see the waves are coming in there. It's pretty, and we just saw a seal. Literally was like, what do you think? Like 20 feet away from us?
Yeah.
Yeah. It can poke its head up and looked at us. It was observing us way more than we were observing it, which was super. I mean, this is pretty phenomenal. Look. So this is where we're at right now. How crazy is that?
lot of scientific activity that goes on here, and we've been able to put some new instruments in one of these sheds. Go have a look. There is a pumping system that pumps seawater up to this instrument room, and there's a tube inside this this material. Seawater that's coming through this line is used for RNA and DNA and protein analysis and in situ physiological measurements. So it's very important that the sample is recovered in a way that enables maintenance of the biological and physiological activity that's occurring. We wouldn't want to sample the seawater in a way that is destructive or inhibits processes that are going on. So that poses some limitations on how the sample can actually be acquired. And phytoplankton are, of course, very, very sensitive to light. At high light, they become stressed, produce reactive oxygen species and other stress. So that's why the sample is dark and kept in the dark until it is funneled into the various sampling devices. So this seawater comes into the shed, and it is used for real-time imaging of the phytoplankton community. It's also can be sampled for eDNA or RNA, and it also can be analyzed for gases that are signatures for metabolic activity like photosynthesis and respiration. So the impact of nutrient delivery events on phytoplankton blooms at a genetic and physiological level can be observed in more detail. So, the line that we were just looking at outside comes here. It's split, and one sample can go over here to this is a membrane inlet mass spectrometer that is for analyzing gas content of seawater and signatures of metabolism. And then also the seawater is delivered into this device, which can be triggered remotely to sample for RNA and DNA, and also into this instrument, which is an imaging flow cytobot, an IFCB, which has been developed real-time automated observation of the microbial plankton community. And so right here we can see in real time the phytoplankton community that's in the seawater. After various forcing events like the delivery of nutrients or a change in temperature or monthly or seasonal changes, we can examine the diversity and size of the phytoplankton community in response to various environmental triggers.
Quite a step up from a bucket.
Right. That's a big dinoflagellate. And then I'll show you, let's see, this is...
All right, you guys, on May 1st, you guys can talk live on the Zoom link at lovestemsd.org to Professor Andy Allen, who will talk to you more about diatoms, phytoplankton, the technology that they're doing it, and a whole bunch of other stuff. Until then, guys, stay curious and keep on sciencing.